Good afternoon, everyone. Welcome to the Wednesday afternoon lecture and to a presentation by a distinguished researcher and a leader in the whole area of science and science policy, Keith Yamamoto. Keith is currently the Executive Vice Dean and Vice Chancellor for Research, as well as Professor of Cellular and Molecular Pharmacology at UCSF. And yet, despite all of those roles, continues to do a really interesting science related to the glucocorticoid receptor, which he's going to speak to us about. He got his undergraduate degree from Iowa State, his Ph.D. from Princeton, and following that, landed at UCSF, which he must have liked rather well, because it seems that that's where he has been, effectively without interruption, but with lots of different positions since 1973. And now, having arisen to this position of Vice Chancellor for Research, has a critical role in steering that remarkable institution at a remarkable time. He has also been elected to the National Academy of Sciences, to the Institute of Medicine as well, and is a fellow of the American Association for the Advancement of Science. But I want to say that Keith is also one of the most dedicated citizens of the biomedical research enterprise in the way in which he serves NIH. He has been on numerous study sections and chaired several of them. He has served us most recently on the biomedical workforce and diversity in the biomedical workforce working groups of the advisory committee to the director. And Keith, I've greatly appreciated your wise advice in those areas. He's currently on the advisory council to the Center for Scientific Review. He has played many important roles in our deliberations about how to make our peer review system, which we think is the best in the world, but it's not perfect, so we continue to try to make it better, and Keith has been very influential in that regard. He also has played a really critical role in terms of trying to get the word out to everybody in the community, various constituencies, about the value of medical research, and he serves as the chair of the Committee on Life Sciences, which is a coalition of scientific society groups that makes that case very effectively. And goodness knows, this is a time where that case needs to be made particularly effectively. So usually when Keith is here, we're having some very intense debate about peer review, about science policy, about funding. But it is a delight to be able to invite him to come to the podium today to talk about science and the science that he does most ably. And I'm sure you're going to enjoy what he has to say. Following his presentation, we'll have time for a few questions, and there are people listening on the web, so we'll ask the questioners to come to the microphone, and then there will be a reception in the library where you can continue your conversation with Keith. His title today is Combinatoriality, that's quite a mouthful, Signal Integration and Allostery, Determinants of Specific Transcriptional Regulation by the Glucocorticoid Receptor. Please join me in welcoming Dr. Keith Yamamoto.
It is great to be here to talk about science for a change, and I really appreciate the opportunity, so thank you. It's nice to see a lot of you here. Francis is trying to disrupt things by giving me a letter just before he came up to introduce me. Harold and I were talking, and he somehow got out of our permanent file a letter in which we were competing over a bit of science back in the 70s. So I can't remember if that's been resolved or not. All right, so thanks. I'm going to talk today about this, and I apologize for this title. I think that anybody who turns in a 3-line title with a colon in the middle is mostly just displaying their insecurity or something. So I apologize for that. But what I want to tell you about is a series of studies that we've been carrying out over a long time in the lab that we're excited about because I think they give us some hints, some clues about metazoan transcriptional regulation that could hopefully will be general and could not only be informative at the scientific level, but give us some insights that could be useful in various kinds of applications, and I'll mention a couple as we go along. And what I want to do is to back way up and to start with something that we actually knew or know something about. I call it the good old days of what we thought about when we were looking at transcriptional regulation in bacteria. It's notable to put this up because it is the basis for a lot of things that we think about, and here at the NIH, of course, tremendous amounts of seminal work in this area was actually done back when, you know, the giants walked the earth with Felsenfeld and Adjia and Nash and, oh, I guess they still walk the earth, actually, come to think of it. And so textbook version was, of course, the lac operon. What we knew then and know now is that transcriptional regulation works by affecting the capacity of RNA polymerase to bind to promoters and to act when it gets there. And the regulation is relatively simple. One or two signal responsive factors that affect the binding activity of polymerase, the familiar lac repressor, of course, and the cyclic AMP binding protein that actually Harold was involved in characterizing when he was here in the lab, in our Passon's lab. And this sort of gave us a way to think about things. I'll just say that the actual mechanisms, aside from occlusion of RNA polymerase binding, but the actual mechanisms by which positive regulatory factors work by touching RNA polymerase and then doing something to make the polymerase work better, still have not been mechanistically solved. We certainly have a paradigm for thinking about what's going on, but I think that in a sense, if one wanted to understand the mechanisms of transcriptional regulation, there's still a huge amount of information to be mined from these simple systems. So where can we go from there? If we begin to thinking about what happens in metazoans and how transcriptional regulation works, we could take this model and look at the regulatory factors that reside in those cells, such as the glucocorticoid receptor, and draw a model that looks similar. So what we know is that the glucocorticoid receptor resides in the cytoplasm of cells in the absence of hormone. It associates with hormone, changes its properties so that it goes into the nucleus, binds to specific sites on the genome, glucocorticoid response elements, GREs, and regulates transcription either positively or negatively. So it looks like the same simple framework we've been looking at, but that we know that prokaryotic-like model can't be the whole thing. It can't be that simple because glucocorticoid receptor action, unlike in E. coli, is context-specific. Cells are different, one tissue type to another. It's not all one E. coli cell after another. And the way that the receptor works in those different settings is actually different. So if you look around, if you walk around this wheel, here's a series of, in this case, tissues or organs in which the glucocorticoid receptor works. And as you read the little captions, you can see that in those different settings, the receptor is doing different things physiologically, and we know at the molecular level as well that that's true. So the same regulatory factor binding to the same small molecule cholesterol-derived steroid hormone is having these very different actions, and it's doing that by regulating different genes in the different cells. So it looks like a real serious departure from what we're familiar with in the case of the lac operon. So the question then arises, how can a single regulatory factor, the glucocorticoid receptor, function in this way so that as you go from setting to setting, what happens in a liver is very precise with glucocorticoids and it's very different from what happens in the kidney. How do you get both precision in stamping out the same set of regulatory genes or regulated networks in a given setting or an organ, and yet be flexible enough so that when you go over to the next organ over, everything changes. And changes are actually rather dramatic in terms of the fraction of genes that are distinct in one setting or another. Now, of course, that's not all. This is not just sort of a party trick for molecular biologists, but in fact, all of these changes are themselves coordinated into a physiologically sensible whole, so you end up having the steroid hormone that's put into the bloodstream signaling a physiologic state that the body can then respond to in a logical and orderly way. So there's a network of information that's being somehow handled in this way so that you end up with precise gene regulation, yet flexible from one setting to another. So it's that paradox in a sense that we'd like to sort of struggle with here. Now, what do we know about what's going on in metazoans, or in fact in eukaryotes in this case? Again, textbook level information, and all hell breaks loose. Things are complicated. You don't have binding sites for 1 or 2 factors, you've got binding sites for many. They're not just sitting right next to the RNA polymerase, they're kind of scattered all over in the region of the gene, sort of. Right? A lot of different factors are binding. At least in some cases, they've been shown to kind of aggregate together in this sort of way. And this is the sort of cartoon that, you know, sends people screaming for the exits. You were free to leave, but you're not. That don't work on transcriptional regulation, and they basically say, you know, when you figure it out, please come back and let us know, but in the meantime, please don't bother us. So the question is there a way to think about this in which we can move forward? And so I'm going to present one that I think is a simplifying assumption that I think is actually based, at least conceptually, in fact, and I'm quite certain that it's based, in fact, in general. To sort of deconvolute this, at least in our thinking, consider the notion that this big glob of stuff is, in fact, can be thought of as a single factor. And so that would say then it's the combination of gene regulatory proteins, that come together, and that combination can be thought of as a single distinct factor. It has multiple polypeptide chains, but a single factor. That is bound specifically to a composite response element, different patches of DNA sequence to which some of those factors associate, to then give rise to this assembled complex that can be viewed as a single distinct factor, and it's this combination that makes a difference. Now, this too is not a new idea. Britton and Davidson in the mid-60s, maybe the early 60s, began to write about the notion of solving the problem of how we gain specificity for regulation by using just kind of combinatorial arithmetic as a way to solve the otherwise endless loop of saying if you propose a cell-specific factor, for example, you need to explain how you make a cell-specific factor, and what you would do is invoke a cell-specific factor. Right? That doesn't help. This allows you to escape that problem. We can talk about it in more detail, but I think it's almost obligate that something like this, this combinatorial notion, is in play. So now you can see that this again looks like a Lac operon, a single factor associating near or next to a promoter, in this case for RNA polymerase II, and regulating its expression in a specific way. So combinatorial regulation, gene, cell, patho or physiology-specific complexes that are built from different combinations of factors that are themselves not unique, but in many cases really broadly expressed, but different combinations, different subsets from a long menu of factors that are produced in a given cell. This is nice, but of course it raises then the next level question. What specifies the context-specific assembly of this unique combination? So it's great that you can invoke a unique combination. What specifies that? And I'm going to give you 2 levels of answer to that question, or 2 ways at least to close in on a way of thinking about what the answer is. Oops. Sorry. So the first is this combinatoriality. But to answer the second question is what specifies this assembly of a unique combination of factors is the second level answer, and that is that it's signaling. That's signal, in fact, specifically signal integration. The integration of multiple bits of incoming signaling information that, in this case, impinge on a single regulatory factor. Here's the anatomically correct version, description of the glucocorticoid receptor from N terminus to C terminus. AF stands for activation function, so these are transcriptional regulatory domains on the protein. There's a DNA binding or genome binding region in the middle, and at the C terminus is the region that associates with the hormone. So I say that there are multiple signals that are impinging on this. Well, of course, we know one of them because it's the hormone. And so I think the interesting thing about these multiple signals that I'm going to recount for you, and I'm going to talk about four for classes of signals, is that in aggregate, they provide signaling information to the receptor that accounts for this need to be gene, cell, and physiology specific. The ligand, of course, is a physiologically specific signal, right? There's a neuroendocrine signal that says make cortisol. It drives the signal to the adrenal cortex. Cortex dumps cortisol into the bloodstream and says the physiological state of this organism is such that it's under stress. It's trying to give a seminar at the NIH, so make more glucocorticoids. So it does that. So here's a physiological status signal for the receptor, and it binds in its determinants. The receptor also binds DNA. Specifically, it binds to what we call a GBS, a GR binding sequence. That sequence is a big set of variants of this 15-mer, which you recognize has palindromic hexamers that are separated by 3 base pairs. So the GR binding sequence now, of course, is a gene-specific signal. This is the signal that's providing the receptor with information about where it is on the genome. It's binding specifically to the sequence. The GRE that I refer to, the glucocorticoid response element, is a region of the genome that's able to confer a particular flavor, context-specific flavor of the glucocorticoid response. It embodies the GBS, but that's not all. So what a GRE is a larger region of the genome that includes not only the GBS sequences, but binding sites for other non-GR transcriptional regulatory factors as well. So every GRE is a composite element that contains binding sites for the glucocorticoid receptor and for other non-receptor DNA-binding transcriptional regulatory factors. So the GRE and the GBS then are gene-specific signals. They say to the receptor, here's where you are on the genome, right, and behave accordingly in some way. So somehow the sequence is conferring some signaling information as well. Now you recognize that the factors that then associate are going to give some cell-specific, cell context information to the receptor, because it's going to be the menu of factors and the levels of their activities that are in fact cell-specific. So that while there are very few factors that are made on only one cell type, the relative levels of the menu of factors does vary in cell-specific ways. And so the available and levels of activity of the non-GR factors will provide cell-specific information to the receptor. And finally, the fourth class is post-translational modifications. Receptor itself is modified in a bevy of different ways, and each of the 7 or 8 or 9, I guess now, phosphorylation sites, for example, on the receptor, is an endpoint of at least one signal transduction pathway, signals coming in from outside of the cell that will then activate a kinase that eventually, a kinase that reaches the receptor and decorates a specific serine or threonine residue. That's just one example. It's one kind of signal. So each of those, then, is providing physiologically specific signaling information to the receptor by decorating specific sites along the way with particular chemical moieties. So now you can see that if you then do the combinatorial arithmetic on this, the number of combinations that could be in aggregate being input information into the receptor to say, here's your status, physiologically, cell-specific, and gene-specific, and respond accordingly. So that is the idea then, and it's having done that then, that co-regulators then associate and begin to build out a regulatory complex that is built of many proteins, probably on the order of 100 polypeptide chains in a grown-out specific transcriptional regulatory complex. Now there's lots of action, much of it, some really exciting work here at the NIH on the dynamics of these complexes. So the assembly is one thing, but what we know is that the ways that they're produced, how stable they are, the necessity for deconstructing these complexes once they're built. There's a lot of action here about which there's a growing body of information. As I said, some of it collected here in the Hager lab, in the Larson lab, and others that are going to be very important. I'm not going to address any of that today, but it's important to know that the cell is not a thermodynamic entity. And there's lots of kinetic activity that is going to need to be considered in the way that we think about these complexes and how they work. So they're not just stable blobs on the DNA the way that molecular biologists like to draw them. Okay, so now we have at least a way of thinking of saying the receptor can be sort of made different in very unique ways that respond to physiology, cell type, and gene to then be able to do different things. Well, how does that actually work? And the hypothesis, of course, from the title of the talk, is that the signals are allosteric effectors. They're changing the shape or the conformation of the receptor in specific ways to then drive the assembly of unique complexes of factors. So that's the notion, that integration of allosteric signals could produce GR surfaces that recruit distinct combinations of factors, right? And that kind of drives your thinking in a couple of interesting ways, or at least drives my thinking in a couple of interesting ways. First of all, it says that the regulatory complex composition and activity and structure depend on these different physiological contexts. There's nothing hardwired about the shape of the receptor and what it's going to associate with. And then the second one's important too, and that is the signals, and therefore the receptor itself, but in this case the signals, lack intrinsic activities. And so if you open a pharmacology textbook, you'll be able to look at a set of structures that say, oh, this is a strong agonist, this is a strong antagonist, and so forth. That's true in some contexts, but that strong agonist in a different context may be an antagonist or a weak agonist, right? So it's all context specific, and this notion that you can identify things with a given kind of activity is true only in a given defined context. So I'm going to spend the rest of the talk then talking about these two questions, very asymmetrically, actually. One is that with these notions in place, how can we think about how to achieve this precision and flexibility? All right? And I'm going to scrunch, actually, one postdoc's a huge amount of work from Shenghong Chen in the lab into 4 or 5 slides to summarize that. And then spend the rest of the time on the second question of this idea, the hypothesis, that the signals are acting in this allosteric way, changing the shape of the receptor and therefore specifying the surfaces to which other factors can append, and then building out specific regulatory complexes from there. So, Shenghong Chen, fantastic postdoc, came to the lab and said, this precision flexibility thing is cool. Let's do something to figure it out. And his idea was, he came to the office with an idea that I thought made no sense to me. And he said, what we should do is refine the question. So we can look in 2 different cell types at a gene that's glucocorticoid regulated in both cell types, but by different mechanisms. And I said, you're right, Shenghong, that'd be great, a simplifying way to look at things, but how can you look at a gene that's regulated by the same factor, GR, in 2 cell types and say this was regulated by GR by a different mechanism than in this cell? And he said, not a problem. He actually went away and came back, said, not a problem. We'll look for genes that are activated by GR in one cell and repressed, and that same gene repressed by GR in another. And I said, I knew that. I was just trying to figure out if you would think of it. So he sorted through our expression arrays, and in 2 cell types, human cell types, A549 from lung, U2OS from bone, osteosarcoma, actually. And out of the several thousand genes that are glucocorticoid regulated in those cells, I think about 2,000 of them overlap, and out of those 2,000 overlap, 31 were regulated in opposite directions. So here's an example of 4 of them. And you can see they're, in this case, repressed in U2OS, activated in A549, and the opposite here, and so forth, looking across the time course. So that was interesting. So he had a set of things to look at, so then he could ask, where does the flexibility come from to be able to kind of switch mechanisms like this in this sort of profound way of looking at positive and negative regulation? How are precision and flexibility achieved at these so-called poised GREs that can either end up on one side of the fence or the other with respect to activation or repression? And when he began to characterize these genes, he got a surprise with one of them. It's cartooned here. So here's dose-response studies, and you can see that there's some aberrant behavior. So the dexamethasone concentration at which he picked up these 31 genes was 10 to the minus 7th molar. And you can see activated in 549, repressed in U2OS, and vice versa, at the standard concentration of dexamethasone. But when he did the dose response, look what happened. This gene, anchored 1, goes through this discontinuity. And so the gene is indeed repressed at 10 to the minus 7th molar at the standard concentration. But at lower concentrations, it's activated and then crashes and becomes repressed. So we puzzled over that. And Sheng Hong said he didn't know how to think about it, but what he was going to do was to try different ways to vary the levels of activity of the receptor. So he wasn't thinking of this as the low dexamethasone concentration. He was thinking of it as a way to achieve a relatively low level of GR activity in the cell. And he said, we've got other ways to achieve that. We have other ligands that aren't as strong. We have, at least in some contexts, we have cofactors that the GR depends on that we can play with. We could look at receptor levels. We can put in mutations. And so he basically did all of them. And so that's what he did. He wanted to modulate the level of GR activity by changing all this other stuff. I'm not going to drag you through that, just to show you one cartoon that makes an important point. So now you're looking at this two-dimensional heat map. I know you can't read the labels from beyond the first row, but this is time and hormone dose. And you can see from the shape of the two-dimensional heat maps that each of the 4 genes is different. But more importantly, what you can see is that there are conditions in which at low levels of activity, each of the genes is activated and then repressed. If it's not repressed down to the baseline, it ends up being net activation. If it's repressed below the baseline, it ends up being net repression. But all of them display the same discontinuous behavior. So what that told Sheng Hong is that there might be a constant transcriptional regulatory motif that is stamped out for each of these genes. Now, whether there's something fancy about the GREs at these poised genes or not, don't know. I'll bet not. But at least in each of these cases, he could find these conditions where he changes the level of receptor activity and ends up with this surprising behavior. Well, it's surprising unless you're Uri Alon and have been studying transcription network motifs, because that's what he'd been doing for a decade by the time, almost a decade by the time Sheng Hong did these studies. And he published in 2003 a series of these transcription network motifs, behaviors, and that what Sheng Hong noticed is that all 4 of these genes, each of these genes, could be, their behaviors could be accounted for with one such network motif that Alon had given this charming name of incoherent type 1 feed-forward loop. Really rolls off the tongue, I think. And what characterizes this type 1 FFL is this, and that is that at low regulatory factor, Alon did these studies in bacteria and then in yeast, and then more recently in eukaryotic cells, metazoan cells. But at low levels, in this case of GR, the target gene is activated. But as GR activity increases, it passes through an undefined, molecularly undefined mode called an inhibitory threshold, where the receptor switches from being an activator to a repressor. And so at a time or dose or something affecting receptor activity dependent way, the gene switches from being activated to being repressed. So, signal-specific effects. So for different signals, different genes are affected in different ways, giving rise to these different patterns. Signal-specific effects on activating and inhibitory arms of this network could produce precision and flexibility and give rise to gene activation, gene repression, or in fact, no net activity if they're balanced. So there's two ways to not have a responsive gene. One is to balance activation and repression, and the other is to not have the receptor in play. And so by affecting the relative amount of attention that each of these arms gets, you'd end up with a condition or context-dependent activation or repression. In the case that we were looking at, cell-type dependent. Now, this notion has interesting implications for the composition and structure and activities that one might find in regulatory complexes as you look on one side or the other of this activation versus repression notion, and those are the kinds of experiments that are now ongoing in the lab. Okay, so the kind of most provocative, I think, notion out of this, and one that we're certainly excited about and operating on right now, is this notion that this signal-regulated feed-forward loop network machinery might actually just be resident within the regulatory complex itself. And we have various kind of indirect experiment reasons for believing that that is the case. If you read Alain's papers, he has the different arms of the network actually being activation of different genes. Direct activation of the target gene and then activation of another gene that then becomes a repressor on the target gene. That works too, but we've done experiments that suggest that it may not be that complicated, and that a more elegant solution would be that the switch is actually residing within the regulatory complex itself. And my bet is that the molecular changes that are involved in throwing that switch that provides the flexibility may actually be quite simple. We'll see. But that's the idea, is that this set of network motifs, and there are others that have been at least inferred, if not identified, in the case of GR, other than the incoherent type 1 feed-forward loop that we're looking for. So, sorry. So it could then be that signal integration by regulatory complexes themselves could confer this precision and flexibility. Okay, so I'm going to leave that, and I'll come back to it at the end, and move on to the second mechanistic question, and that is the notion of GR allostery driving this regulatory specificity. That notion had been around the lab for a long time in various forms, but a set of experiments by Lisa Watson in the lab got us on track to begin to do some direct structural analyses. This really took off on it was a sort of a takeoff on some experiments that had been done earlier by Seb Meising in the lab, and I'll show you some of his direct contributions in a moment. Simplistic in the extreme. Take a 15-base paired GBS sequence, take a half a dozen different ones. Those of you who are close enough can see that these aren't very different. One hexamer is always the same on the left. The spacer sequences vary a little bit, and the right-hand hexamer varies a little bit. That's it. So they're very similar 15-mers. They're tacked one element into a simple reporter plasmid and transfected into cells. And Lisa asked two questions. One is, are sequence variants of those 15-mers enough to lead to differences in transcriptional regulation? You can see that they are. Activity varies over a four-fold range when all you've done is to change the spacer sequence. The spacer sequence is almost 20 angstroms from the nucleotides in the spacer, or almost 20 angstroms away from the nearest part of the protein. Change the spacer sequence, and you can get a four-fold variation in the level of gene activation. So the sequence itself is doing something important, and we'd like to know what it is. The key second experiment that really said that she should think about structure or conformation is that there's not a correlation between the transcriptional regulatory activity and the affinity with which the receptor binds to these sequences. In fact, it's almost the opposite. So the most tightly binding ones regulate the least well and vice versa. So something else is going on that's interesting. GR activity does not correlate with GR affinity. So that got us thinking about structure. Now, we've been thinking about structure for some time, and Seb Meising, the aforementioned, and Miles Pufall had done some experiments done and published some experiments a few years earlier that helped to sort of drive the experimental directions that Lisa Watson pursued. What Pufall and Meising had done was to crystallize the DNA binding domain, just a little piece of the receptor, about 10% of it, 89 amino acids, onto different 15-mers, and then ask if the conformations, the crystallographic conformations of those receptors were different when they sat in different sequences. And the answer was sort of one of these sort of answers. And that is that there was one region of the DNA binding domain that we call the lever arm for historically forgettable reasons that changed in its conformation, and only that. So it was sort of like a half answer. What do we do with this answer? This is, again, the lever arm is far from the DNA. So how is it getting information? We're trying to invoke allostery. Shouldn't there be some changes here that would then, what's going on? We knew from earlier crystallographic analyses, and it was confirmed by Pufall and Meising, that only 6 nucleotides of F15 were actually contacted in a nucleotide-specific way by the receptor. So these guys in red are actually touched by this recognition helix as it reaches into the major groove and touches the edges of bases. So, does the lever arm conformations imply some sort of allosteric path? Question mark. We played it up big in the science paper, but we didn't know. And what's going on? Only 9 of the 15 GBS nucleotides are not contacted, and we already knew from Lisa's experiments that she could change nucleotides in different GBSs that were not touched by the receptor, and yet the transcriptional regulatory activities were changing. So both of those questions, very relevant. Now, from staring hard at the crystallographic work, Miles and Lisa were able to draw one conclusion from one pair of GBSs about what could be going on as you change the spacer sequence, and that's this pair. And again, those of you who are close enough to see will see that the hexamers are identical in these 2 GBSs, and that all that is different is that the spacer changes from G's to A's. Now, we knew from old work, maybe Felsenfeld was involved in some of this, or Andrew Travers and others, that had shown that when you have strand bias like this and have A's on one strand, you tend to constrict the minor groove. And the crystallographic structures show that that was true. So this gray guy that has a run of A's has a narrower minor groove than the one that has the G's. So that was interesting because it indicated that the spacer sequence, or just confirmed that the spacer sequence was actually changing the DNA shape. And a hard look at the side chains that were in that neighborhood revealed that there was a lysine residue at 490 that flipped on whether the minor groove was narrow or was normal or narrow. When it was normal, the side chain came down and made contact with this strand of the DNA. And when the minor groove was narrow, the side chain flipped and reached over and made contact with the other strand. We could put together a hand-waving argument that argued that when the side chain flipped, it would affect the conformation of the lever arm. That's fine, maybe that's the case. But at least in this case, we could argue with some reason that the DNA shape was being read by the spacer sequence, or was being affected by the spacer sequence and then being read by the rest of the receptor in some way that might involve this flip. So that's all nice and gives us a way to think at least about this. The spacer sequence changes here. We don't have good ways to think about the other ones in the analysis that we've done. So it remains an open question. But it left still open the even bigger question of, is there any evidence for some kind of allosteric path that would go from that helix reaching into the major groove and touching the edges of bases and sensing the sequence differences, and then driving out to conformational changes in the rest of the DNA binding domain that could then transmit information to the rest of the receptor. So Lisa said, well, something that we were actually worried about when we started the crystallographic analyses is that we were probably going to be looking for conformational changes that required such small changes in delta G that it would be easy to imagine that crystal packing forces could mask those changes, and maybe that was why we were seeing changes only in the lever arm. So Lisa went off to learn NMR. So what you're looking at here is a two-dimensional heteronuclear single quantum correlation HSQC spectrum by NMR. And if you don't know anything about NMR, all you need to know is that what HSQC is scoring is the chemical environment of the amide bonds at each amino acid in this 89 amino acid GR fragment. There's 89 spots here, and each of them is reporting on the chemical environment splayed out in 2 dimensions of the amide bond at each amino acid. Lisa went to the hard work of assigning each of those spots to a specific amino acid, and she could therefore ask, how does the binding sequence affect the conformation? So what you're looking at here is just the GR DNA binding fragment alone. So what happens when you put it onto DNA? What happens when you put it onto different DNA sequences? And that's what's shown here. So now you've seen the overlay at 3 different sequences, and each of the colors reflects a different GBS, and you can see, even from the back of the room, that some spots are completely coincident and that others are not. I've boxed 3 of these positions because I'm going to be talking about them as we go through the rest of the talk. Positions 477 and 478 are in the dimerization region. We're going to look harder at that. And position 470 is in the lever arm. So already we can begin to extract information from these by asking what GR surfaces are being affected, by referring the NMR structure back to the crystal structure to see where we are in the molecule as it's sitting on the DNA. So for example, Lisa was able to learn quickly that changes in the spacer, here's a set of them, different spacer sequences, but not in half-site positions 13 and 15 that we chose for specific reasons that they're not contacted directly by the GR. Changes in half-site position 13 and 15 affect the dimer interface. The spacer sequences, as you change spacers, you can see that the spots are in different places for 477 and 478. But as you change the half sites, they're not in different places. So she can say, if you change the spacer sequence, you change the structure in this region of the DNA binding domain. But if you change the half site, you don't change the structure in there. There's some specific map that relates changes in a particular part of the GBS sequence to changes in conformation. Not only that, by looking at, in this heat map representation, looking at the residues that change in their conformation as a function of the GBS sequence, she can now see that there were changes that extended from the DNA reading helix all the way through the lever arm and out to the dimerization region, and that she could at least infer an allosteric path. So the DNA signals then appear to be transmitted from the DNA reading head out to at least 2, if not through, the dimer interface. And it led to this functional question then, does that matter, right? Does the dimer interface contribute in some way to the specificity of transcriptional regulation by the receptor? So the way to test that is to make a mutation in the dimer interface and ask what the consequences are structurally and with respect to activity. So Lisa did that, and she chose this residue for a particular reason, and that is that mutations at that site, and so in 1991, when we crystallized with Paul Sigler the DNA binding domain of the receptor on a generic GBS, we were able to see a set of 4 contacts that brought these two monomer domains together. And one of those sites, A477T, got famous because Günter Schutz made a mutation in them and has carried that mutation through lots of different physiological settings in cells and now all the way into mice, and is able to see phenotypes all the way at the animal level of changing that residue. So we knew that it was a functional residue in some ways. Whether it's actually more important than others, no other sites have been explored to that depth. But at least we knew from Schutz's work and work derived by others that have looked at this mutation that something functional was actually occurring when you made this mutation. It was just a molecular biological tool. So this region then makes inter-subunit contacts, and you could ask what happens when you disrupt the dimerization region. How does altering the dimer interface affect GR conformation? And you can see again in the overlay that there are changes. You can see from the heat map that those changes that originated the dimer interface actually then extend all the way back to the DNA interface. So the dimer interface mutation affects the DNA binding interface. Allosteric coupling appears to be bidirectional. Again, thermodynamically, you'd predict that to be the case. And this is sort of genetic support for that idea. I'm not going to talk at all about a beautiful series of studies that Seb Meising has done, looking at there's a natural splice variant in the GR called GR gamma, and it moves a 5' splice site in a way that extends the messenger RNA for the GR by 3 nucleotides. Those 3 nucleotides code for an arginine residue that sits right in the middle of the lever arm. So we were very interested to see what the consequences of that were for structure and activity. Seb has done a beautiful series of studies that again show that when you mutate, in this case, the middle of this inferred pathway, you see changes that go out in both directions that affect both structure and activity. So it's a really pretty series of experiments of his. And of course, this also has broad implications for combinatorial signaling. Now, having this effect go out from the DNA interface out to the dimer interface at least led to a question of whether the allostery actually goes beyond. Where there isn't a beyond, it's just a little fragment of the GR, but there actually is a beyond because it's a dimer. So you could ask, does the signaling from one hexamer that one monomer is contacting go up through that monomer, extend through the dimer interface into the other monomer? That would be interesting. So at least ask that question. Here, just to simplify things, she's looking only at this one residue in the lever arm, because we knew that there's a lot of sensitivity, conformational and functional sensitivity there. So looking at glycine 470, here is a GBS in which the hexamer sequences are different. So it stands to reason that the lever arm residue in the 2 monomers would be in different chemical environments, and you see 2 spots for G470. In this response, in this GBS, the hexamers are the same. So it stands to reason that the G470s in the 2 lever arms in those 2 cases would be in the same chemical environment, and there's only a single spot. That's cool, but that's not the interesting result. The interesting result is here, and that is when you overlay them, this single spot doesn't align with either of the spots here, despite the fact that this sequence is the same as this sequence and this sequence. So the inference is that the spot here is different than these spots are different than these guys because they're cross-informing each other and saying to each other, we have a dimerization situation here where the 2 monomers are in different states and they're feeding information across their dimer interfaces. That's the idea. When Lisa extended this to look at other GBSs, it was supported. So now she's looking at 4 different GBSs, and you can see that there are 4 spots. So in every case, the information is different. So what happens then if she mutates the dimer interface? This region that she's inferring is the feeder of information from one monomer to another. Mutate that one residue, and everything collapses into a new position, not the same as any of the others, right? And it says they stop giving each other information, and when they do that, then the information just collapses to one kind, and the receptor is unable to distinguish different GBSs. So that's the model that in the wild-type dimer interface, the different subunit states are feeding information to the other subunit. In the disrupted dimer interface, you end up with yet a different conformation and a loss of that feeding of information. That the GR 717 mutation fails to transmit the DNA signals to the partner monomer and fails to discriminate between different GBSs. So that dimer interface is critical for defining an allosteric path that actually is amplifying the amount of information that comes from the GBS, because the 2 hexamers half-sites are not only informing the monomer structure that is bound to it, but also the dimer partner on the other side. So then the functional question, again, you can see the way we do these experiments in the lab. The functional question, that's great. Does this 477T mutation affect the activity of the receptor? This is a nice structural construct, and it does. So again, without dragging you through each example, if we just look at these 2, where SGK and SGK-GGG, where the spacer sequence is altered, but otherwise the hexamers, while they're different from each other, are different on the 2 halves, are the same across the 2 GBSs. And you can see that with the wild-type GR, the activity, the transcriptional regulatory activity, is very much spacer sequence dependent. And in the mutation, it loses that discrimination. So the A4770 then discriminates poorly between the GBS sequences, just as the structure had collapsed down to a single structure that was distinct from that at the wild-type elements. I'm not going to dwell on this except to say that Lisa did a very extensive series of biophysical characterizations of the wild-type and A477T mutation. This was in part because there had been a dogma that had built up in the field about this particular mutation that had argued that it fails to bind to DNA and that it loses cooperativity and doesn't dimerize anymore. And we think that it's very clear from Lisa's studies that neither of those things is true. It clearly binds DNA, it regulates, it activates, it represses, it dimerizes, and it binds cooperatively. I won't take you through the gel shift data and surface plasmon resonance studies, except to dazzle you with how beautiful Lisa's experiments are, and to give you the conclusion points that the stoichiometry of binding is the same. You always get dimers with A477T mutation. You lose a lot of cooperativity, but it's still very cooperative. The wild type has a Hill coefficient of 1.7, which is very handsome, and the mutant has a Hill coefficient of 1.4, which is still very good. And so making that one change in the dimer interface and compromises cooperativity, but clearly doesn't lose it. You lose about an order of magnitude in affinity because you increase the dissociation rate by about an order of magnitude. And again, there's very poor discrimination between GBSs and the biophysical characterizations, exactly as we'd seen, Lisa had seen poor discrimination in structure and poor discrimination in regulatory activity. So this just is sort of a final structural cartoon that tries to make the point, as you just sort of look at it from afar at the heat maps, that there is that particular changes in the GBS sequence lead to changes in different regions of the GBS. And so, in principle, one could learn enough to become predictive about this and make specific predictions about the regions of the DNA binding domain that are sensitive to or predicted to change, maybe better said, upon changes of a specific nucleotide pair within the GBS, and whether that will end up being functionally informative, at least in a specific context, is one of the directions that we're pushing. So the GBSs affect the GR DBD conformation at distinct surfaces. Finally, again, return to the structure, the activity question. And really, in a sense, this is a genetic stand-in for something that we're lacking structurally, right? And I told you that we're looking only at this little fragment of the GR, the DNA binding domain. It's a sensible place to start because if you're looking for sequence-dependent signaling, you want to ask what emanates from the sequence, and you start with a reading head. But of course, it's also dissatisfying because we can't say what happens to all those outboard remote domains of the receptor that are so important for forming the presumptive surfaces, or forming surfaces at which we know that at least some factors, and we would infer many factors, associate. So here's a genetic experiment that implies, at least to us, that different GR DNA binding domain conformations correlate with changes in conformation, here read by different utilization of particular surfaces of the receptor in these remote positions. So here's the experiment. Years ago, Inez Rogatsky in the lab constructed a set of cell lines in the U2OS background that contained either the wild-type GR or GR carrying mutations that were clean knockouts of these domains AF1 or the dimerization region or AF2. Simple point mutation here, simple point mutation here, and a triple point mutation here that Jorge Iniguez-Luis had made in the lab some years before. Clean knockouts of those individual domains, leaving the other two domains intact. So now she has stable cell lines that expressed wild type or each of those three knockout mutations, and allowed Lisa and Seb to then ask, what happens with the activity of these, coming from these different GBSs when you make mutations in particular domains? Another way to say it is, how much does the activity coming from a given GBS depend on the activity coming from one of those domains as tested by mutating them, okay? So here's two examples, GBS, PAL and FKBP5, the wild type in the black bar, and you can see that when you mutate the dimerization domain at this GBS, the activity goes up. Mutated here, the activity goes down. Different dependence on that region. The PAL GBS is almost entirely dependent for its activity on AF2. You knock out AF2 and it's dead. Okay? It's pretty dependent in the case of FKBP5 as well, right? Here's an interesting one. AF1, you mutate AF1, PAL doesn't care. It's not using AF1 to mount its regulation here. And you knock out AF1 here and at FKBP5, and it cares a lot. So the PAL R spacer depends on AF2, not on AF1, and the dim mutant is a gain of function. FKBP5 spacer uses all the domains, AF2 the most, AF1 next, and dim about the same. Okay? It implies that the GBSs are putting these outboard domains, the remote domains, also into different conformations. It's being scored as different dependencies of the receptor on those domains for its regulatory activity. Maybe the surfaces form and are utilized very strongly in this case, but not in this case, for example. So the implication then is that this allostery extends from the DNA binding domain into other parts of the receptor, where we and others are struggling with full-length receptor preps and different kinds of structural analyses to ask that question directly. Okay, so where have we been? Combinatorial signaling is the long title. Conclusions, perspectives, and speculations. I'm not going to tell you which is which. Combinatoriality. Distinct combinations of regulatory factors and co-regulators assemble into gene, cell, and physiology-specific regulatory complexes at GR DBD. I can see the slides a lot better here. Regulatory logic. We are inferring that this, from experiments actually largely that I've not told you about, that the regulatory complex machinery may be rigged, optimized, to be able to carry out this precision flexibility paradox. That would be interesting if it was all resident within the regulatory complexes and little changes were enough to flip it, in this case from activation to repression or vice versa. Signal integration, the GR receives multiple classes of signaling inputs, which together produce context-specific effects on GR conformation. And those signal-driven conformations then trigger the deployment, do the allostery. They trigger the deployment of distinct combinations of GR functional domains that in turn provoke the assembly of distinct regulatory complexes. There are other inferences that go beyond this. And in fact, in a sense, these studies, I think, provide one kind of interesting point of access to understanding one of the sort of pillars of biological regulation of being allostery. It's been with us for a very long time. And the detailed mechanisms of how you move structural information, especially through the core of a hydrophobic core of a protein, remain pretty mysterious. And so you can think of then changing the sequence of a GBS as doing ligand chemistry, and pretty easy ligand chemistry, that then allows you to access the consequences of particular changes in those ligands on conformation. And I think it may end up feeding us some information. There's a larger, higher dimension question as well that's interesting. And that is if there is some relationship between the regulatory mechanism in a given place and the physiological endpoint that you are shooting for. So is there some link between physiological networks and transcriptional regulatory networks that we can see? A way of thinking about this is whether one can begin to link regulatory mechanism with physiological output. Now, in bacteria, you can actually do this. You can look at an operon, the arrangement of the regulatory elements in an operon, and say, I'll bet this is a sugar metabolism operon. There is a repressor binding site right next to the RNA polymerase, or blocking the RNA polymerase, and there's a cyclic AMP receptor binding site just upstream. This is going to be galactose, glucose, fucose, you name it. Instead, if you had a regulatory domain that had an open reading frame that read out a series of repeated codons for a given amino acid, and then could extend into the body of the operon itself, you could say, this looks like an amino acid biosynthetic operon. So a link between the regulatory mechanism and the physiological network. I bet that that's going to be the case here, but much harder to see because of all the different signaling inputs. And you wouldn't expect it to match up in this way that you could see by inspection, or at least I wouldn't expect you could see it by inspection. But I'll bet it's going to be there. Right? If that's true, then it at least opens an interesting possibility. And that is that maybe we could find situations, define conditions, where we can reduce complex physiological readouts or substitute, complex physiological readouts, osteoporosis, how's that? Or how about depression? That's not so bad. With a molecular change. Maybe a molecular change in the shape of the receptor, the appearance of a surface, the capability of a given cofactor to associate, a particular modification state that would have activity in one case and not in another. That would change a lot of things. It certainly would change the way that we think about doing pharmaceutical testing, for example. Be free of the vagaries of animals and the time and expense of doing animal assays if you could really validate this in a way that was predictive. This has led us to be on a big search, and I'm not going to go through the disease areas that we're interested in, but for this notion of causative primary regulated genes. Causative meaning that there's a gene whose function or response is essential for the, in this case, hormonal signaling you're looking for. If you knock it out, you lose the hormone response. If you're lucky and it's not only necessary but sufficient, then expressing it in the absence of hormone would give you the phenotype even without the hormone and so forth. You can imagine the tests there. Primary simply meaning, in our lingo, that there is a GRE nearby, a GRE that is not only responsible for mediating the hormonal response, but actually has the receptor bound to it as well, because that's our molecular leverage into the system. So we're looking. I think there will be this link here, and if it's true, it would be quite informative. Okay, so experiments done by a fantastic, wonderful group of colleagues, Chenghong Chen, Samantha Cooper. I didn't tell you specifically about the bioinformatics input of these two wonderfully talented graduate students, Sam Cooper and Ben Schiller, that did a lot of the bioinformatics analysis code writing. Lisa Watson, Sebastian Meissing now in his own lab at Max Planck Berlin. Miles Pufall now in his own lab at the University of Iowa. I'm not an NMR expert by any stretch of the imagination. I found I could spell NMR, but John Gross is at UCSF and has given us lots of advice. And Chris Kuchenbecker, who is a postdoc with Bob Frederick in the lab, collaborated with Lisa to do the surface plasma and resonance studies. And I thank you for all your attention.
Thanks, Keith. I know we're a little past the hour, and people who have to go should certainly feel like you can jump up, but I'm hoping maybe there might be one or two questions for those who have the time to stay and engage. If you have a question, we have microphones in the aisles so that people who are listening on the web can hear the conversation. Yes, go ahead.
So for the complexes that you mentioned in the different physiological contexts, have you or anyone else done, for example, mass spec analysis to look at all of these different complexes, GR complexes in different contexts?
Have we looked at you mean just look at a given particular complex in different contexts? Yeah, I think a lot of people are working on that. We certainly are. And we're looking in particular cell states and then a couple of disease states. We're putting some focus right now on the lymphoblastic leukemias, diseases, big childhood cancers, both T cell and B cell, that are very well treated with glucocorticoids, except they become resistant. And so we're looking to see in the sensitive resistant cases what the changes are in the regulatory complexes.
You said something about there being perhaps as many as 100 proteins involved in these transcriptional complexes that include glucocorticoid receptor. Where does that number come from, and how solid is it, and how variable is it?
I sort of made it up. Is that a problem? So we know of various histone acetyltransferase complexes, chromatin remodeling complexes that contain on the order of 20 proteins. So it adds up fast. Whether you actually have a complex that has 100 polypeptide chains sitting there, don't know.
Sounds a little scary. Well, for those who'd like to continue the conversation, Keith will be in the library where there will be also refreshments, so please come and join. And thank you again, Keith. Let's thank our speaker one more time.